6 is all about cellular respiration, how organisms release energy through metabolic processes. Panda eats the bamboo, and the tree makes its own food for photosynthesis. But what is the food used for? Both require cellular respiration. Molecules in food, including glucose, produce molecules of ATP. ATP is the energy carrier or energy currency used to power cellular activities. Cells need ATP to survive and perform certain functions. You can see the chemical structure of ATP at the bottom here. You got 3 phosphate groups, the adenine group, and the ribose sugar. Aerobic cellular respiration is used by all plants and animals, as well as microbes. Aerobic is in the presence of oxygen. Energy from glucose is extracted, and then that energy is put into ATP. Cells use that ATP for various processes. Cells need ATP. Cells use ATP formed during cellular respiration to do work such as active transport, to move molecules against a concentration gradient, and power chemical reactions. The overall chemical reaction for aerobic cellular respiration, and that's in the presence of oxygen, is the exact opposite of photosynthesis. As you can see, the products of photosynthesis are glucose and oxygen are now the reactants of cellular respiration, and glucose and oxygen recombine to yield carbon dioxide, water, and ATP. We get oxygen from our air. We need it for anaerobic cellular respiration in our cells. So anaerobic cellular respiration would be without the presence of oxygen. We exhale carbon dioxide in the air. That's a waste product from our lungs. It is produced in cellular respiration, but our bodies have no use for it. Plants, on the other hand, require carbon dioxide to run photosynthesis. So in this and many other ways, animals and plants complement each other. Do plants carry out cellular respiration? No, photosynthesis has the same function in plants as respiration it has in animals and microbes. B, no, their energy needs are too small to require cellular respiration. C, yes, they require ATP like any other living thing. Respiration generates ATP. D, yes, they require cellular respiration, such as a way to get rid of extra carbon dioxide. The answer is C. Their cells require ATP to run their metabolism of their cells. During the daylight hours, they're just performing photosynthesis at a faster rate than cellular respiration. Okay, cellular respiration is related to breathing, and photosynthesis is also related to breathing. Cellular respiration releases energy from glucose in a series of small chemical steps. If the energy released at once, much of it would be lost as heat. So it's a very controlled process that has several chemical steps in it. The overall chemical reaction is what we see for cellular respiration. But there are many tiny intermediate parts that you can see in the flowchart here that we're going to break down today. We're going to break up cellular respiration into 3 major steps. Glycolysis is step 1, the Krebs cycle is step 2, and then the electron transport chain is step 3. Glycolysis, lysis means splitting, glyco is glucose, so splitting of glucose. 1 molecule of glucose is split into 2 3-carbon molecules of pyruvate. You can see here's our simplified version of our 6-carbon glucose, and here are 2 pyruvates. So everything is accounted for as we go through because of the laws of conservation of energy and matter. Nothing is lost, it's just chemical reactions change their form. This first process releases 2 molecules of ATP. And notice you also really often see that ATP in the lightning bolt as to indicate It's a molecule of energy. It's the currency of the cell, the energy currency of the cell. Okay, so the two pyruvates go on to be converted into two molecules of acetyl-CoA, as you can see here. Those are two carbons. And then two of the leftover carbons are released as carbon dioxide. Okay, now we're inside the mitochondria. The acetyl-CoA molecules are disassembled during the Krebs cycle. Notice the Krebs cycle, like the Calvin cycle, repeats itself on a rotating basis, assuming that there are molecules and energy feeding into it continuously. Energy from acetyl-CoA is transferred to electrons. These reactions release two molecules of carbon dioxide. Electrons are loaded into the electron transport chain, and this is where we're going to see the big payoff of ATP. So for glycolysis, we get two molecules of ATP. For Krebs cycle, we get two molecules of ATP. And down here is our electron transport chain. It's just an electron pump. An ATP machine, it makes 34 molecules of ATP. These reactions require oxygen and release water. Also happen in the mitochondria. Okay, what happens to glucose's carbon atoms during the overall process of aerobic respiration? They are donated to oxygen. B, they remain in the pyruvate molecules. C, they become part of ATP. Or D, they are released as CO2. And the answer is D. Glucose's carbon atoms are released as CO2. Okay, follow the energy. What happens to the energy from glucose during each stage of cellular respiration? So you should be able to look at the flowchart from glycolysis to Krebs cycle to the electron transport chain and interpret it and track how much ATP is made from each stage, where the carbons go, what if they leave as carbon dioxide, or if they become part of an intermediate carbon chain that goes through the process. Mostly we're concerned with the manufacture of ATP in this process. Cellular respiration takes place in the cytosol and then in the mitochondria. Glycolysis reactions occur in the cytosol of cells, and you can see the cytosol here. The rest of the reactions depend on the cell types. For prokaryotic cells, which are ancient one-celled bacteria that have no nucleus and no membrane-bound organelles, Krebs cycle occurs in the cytosol only because it doesn't have a mitochondria or any other organelles for it to occur in. The electron transport chain occurs in the cell membrane. But we are mostly concerned in this class with eukaryotic cells. And so we're going to look at what happens to the Krebs cycle and electron transport chain inside the mitochondria. So you can see that the mitochondria here has an outer membrane, and the inner membrane has many, many folds, which means that it has a lot of surface area. It's a small organelle, but lots of things can happen here as it has many, many cristae folds for these processes to take place. So these are phospholipid bilayers for the outer membrane of the mitochondria and the inner membrane. There are lots of enzymes in there, and enzymes, remember, are protein catalysts. They lower the activation energy of a chemical reaction that happens in the body, and they make it a lot more likely to happen because the reactants don't have to get to such high energy levels to happen. Especially with the electron transport chain, this is really important. We've got massive amounts of ATP being made. It has to happen quickly and efficiently. And a lot of the enzymes for the mitochondrial processes occur, like there are ribosomes in there that make them right inside the mitochondria. It's a fast-paced process. Between the mitochondrial membrane is an intermembrane compartment, the mitochondrial matrix, which is kind of like a maze, but a very structured one. which houses the enzymes of the Krebs cycle. And this is a close clip. You can see the phospholipid bilayer of the outer membrane, and then yet another phospholipid bilayer of the cristae, as in our blunt view here. And then the cytosol is right outside the mitochondria. Where is the mitochondrial matrix? Outside the outer membrane, between the inner and outer membranes, or inside the inner membrane? The mitochondrial matrix is inside the inner membrane. Okay, so cellular respiration happens in the cytosol for prokaryotes, and then the glycolysis happens in the cytosol for eukaryotes, and then the Krebs cycle electron transport chain for eukaryotes occurs. Okay, glycolysis produces 2 molecules of ATP. Remember that it means like breaking down glucose. Glycolysis, lysis is to break apart. Glyco stands for glucose. It occurs right outside the mitochondrion in the cytoplasm. There are enzymes that facilitate this process. Some extract potential energy stored in glucose. You can see what's going into glycolysis is our glucose molecule here, this 6-carbon chain, 2 NAD plus 2 hydrogen ions, 2 ADP plus 2 phosphates. The net output is the 2 pyruvates, which are 3-carbon chains, NADH, which is a charged electron carrier, and 2 molecules of ATP. Okay, glucose is activated via this first process of glycolysis. It requires an input of 2 ATP to activate glycolysis. The activated glucose is then converted into 2-carbon intermediates called pGal. This is the chemical structure of glucose here. You can see a simplified version in the 6-carbon chain. ATP, when it's being used up from a charged battery of ATP, goes to a decharged battery of ADP, so that's indicating that the ATP is being used up. But it is a rechargeable battery, so that ADP can be recycled. We're looking closer at the steps here. So from glucose, it doesn't go straight to the 2-pyruvate. There are intermediate steps here. Glucose 6-phosphate, so we add a phosphate to the 6 carbons, and then we have fructose 6-phosphate once it's rearranged, and then a second phosphate is transferred from ATP, so then we get this another intermediate of fructose 1,6-biphosphate. The 1 and the 6 tell us which carbons the phosphates are added to, and it's biphosphate, so 2 phosphates. And then we get a 6-carbon intermediate, which splits into 3-carbon intermediate, which is phosphoglyceraldehyde, or pGal. One of the 3-carbon intermediates is converted to the other type, so there are 2 molecules of pGal. Each pGal molecule proceeds to the energy extraction reactions of glycolysis. So the energy harvest part. So we put a 2 ATP in. Let's see what comes out. So then we have oxidation and phosphorylation, and we get 1,3-phosphoglycerate, and we have our first yield of ATP. The ADP becomes recharged as ATP, and then we are left with 3 phosphoglycerates, and then the 2 phosphoglycerates, once they're rearranged, the removal of water leaves us with phosphoenolpyruvate, or PEP, and then we get a substrate level phosphorylation yields ATP and 2 molecules of pyruvate per glucose. So during glycolysis, NADH is produced as well. First, each pGal is oxidized, producing 2 NADH molecules. NADH, we saw NADPH with photosynthesis. NADH is also an electron carrier molecule. It just lacks the phosphate here. It carries electrons that store energy. So then pGal donates its phosphate groups to ATP, producing ATP molecules. This is at substrate level phosphorylation or phosphorylation. Okay, glycolysis. ATP is produced from ADP. An enzyme transfers phosphate from a molecule to ADP, and it's in effect recharges the battery from ADP back to ATP, our high energy molecule. 4 ATP are produced, but we say that 2 ATP are the result of glycolysis because we needed 2 ATP to start the reaction. So the net yield is 2 ATP. Glycolysis is anaerobic. Anaerobic means that it does not require oxygen. Glycolysis can occur in anaerobic conditions. I had to go let my kitten in. You can probably hear her in the background. Glycolysis can occur without oxygen. That makes it an anaerobic process. Okay, to summarize glycolysis, glucose requires 1 molecule of glucose, 2 NAD+, and 2 ADP. Glycolysis yields 2 pyruvate and 2 electron-carrying NADH molecules, and 2 ATP. If 8 glucose molecules enter glycolysis, the net products will be how many pyruvate molecules and how many ATP molecules. Okay, so this is for 8 molecules. So what we've talked about so far was for 1. So for 1 glucose molecule, we need 2 ATP4 to get the process started. And then we have a yield of 4 minus 2, so a yield of 2. So 2 to start, 2 is our net yield, times 8, that would be 16 to 16. Okay, so you'll need to know the starting materials and end products of glycolysis. Aerobic respiration yields many ATP. Okay, so Krebs cycle and the electron transport chain require oxygen gas, and they, as a result, have a much more higher yield of ATP than glycolysis. And that's the process we're going to look at now. Okay, so there's a transition step between glycolysis on into the Krebs cycle. We have 2 pyruvate molecules. From glycolysis, they undergo an oxidation reaction, and they enter the mitochondrion. This is sometimes called the transition step. You can see our 2 pyruvate, our 2 3 carbon sugars going into the mitochondria. The carbon atom is stripped from each pyruvate and leaves the cell as a carbon dioxide molecule. At the same time, NAD+ is reduced to NADH. Okay, so Krebs cycle overview. During the Krebs cycle, the 2 acetyl-CoA molecules are oxidized, yielding 4 CO2, 2 ATP, 6 NADH, and 2 FADH2. This is a new molecule, the FADH2. It's another type of electron carrier. The Krebs cycle, just like the Calvin cycle, is a rotating, loops back on itself. We're going to look at the names of the intermediate steps. but not the chemical drawings of it, as we would look at more closely in a biochemistry class. Acetyl-CoA combines with a 4-carbon molecule called oxaloacetate, and it yields a 6-carbon citrate. So we're going to follow the arrows around and look at what's made. Citrate is rearranged and oxidized and has several intermediates. For each turn of the cycle, these reactions give off 2 molecules of carbon dioxide, as you can see output here. As citrate is converted, each turn of the cycle produces also 1 ATP via substrate level phosphorylation. Someday I'll say that right. As citrate is converted, each turn of the cycle transfers electrons to different electron carriers, such as 3-NADH. Sorry, my kitten. It's driving me nuts. 3-NADH and 1-FADH2. Citrate is converted into isocitrate, alpha-ketoglutarate, succinyl-CoA, fumarate, malleate, and then oxaloacetate. This allows the cycle to start again. So let's look at the ATP produced. Aerobic respiration of 1 glucose molecule has yielded only 4 ATP. But 10 NADH molecules have produced, as well as 2 FADH2. These molecules carry electrons to the electron transport chain. NADH and FADH2 donate their electrons to the electron transport chain, where energy from the electrons is used to produce many ATP. And you can see in this process here, it's kind of an assembly line, if you will, where we're really just starting to crank out the ATP through this assembly line type process. As electrons travel through the electron transport chain, carrier molecules use potential energy of the electrons to transport hydrogen ions into the intramembrane compartment. These hydrogen ions move down the concentration gradient from the intramembrane compartment, which you can see is right here, but blown up, and they pass through an enzyme called ATP synthase. This enzyme ATP synthase produces ATP. This process is called chemiosmotic phosphorylation. It's highlighted in this box here. Aerobic respiration yields many ATP. 1 molecule of glucose through the electron transport chain, previously through glycolysis, then Krebs cycle is 34 ATP. ETC, or the electron transport chain, requires oxygen. At the end of the transport chain, electrons are donated to an oxygen atom, which combines with hydrogens to form water. Oxygen is the final electron acceptor. Without it, the chain shuts down. Okay. So you need to know NADH and FADH2 power ATP formation in the electron transport chain. Okay, cellular respiration of 1 glucose yields 36 ATP. Glycolysis and Krebs cycle each produce 2 ATP. The electron transport chain produces 34 ATP. Transporting NADH into the mitochondria requires 2 ATP, making the total production of ATP equal to 36. That each, okay. So 4 ATP are made in glycolysis, but 2 are required to get the process started. So we have a yield of 2. Krebs cycle produces 2. Electron transport chain produces 4, but 2 ATP are required to get through the membrane of the mitochondria. So we got a total of 36. Okay, other carbohydrates enter energy extraction pathways. Polysaccharides from our diet, like glycogen and starch, are composed of glucose. They just have to be broken down. And that's what your body does in the digestive process. So we can take proteins, starch, and glycogen and fats and break them all down into glucose. Breakdown of simple molecules to pyruvate and acetyl-CoA accompanied by production of limited ATP and NADH. Okay, proteins and fats are also energy sources for the cells. So fats can be directly converted to pyruvate or acetyl-CoA. Amino acids from proteins can be converted to pyruvate, acetyl-CoA, or Krebs cycle intermediates. So you can see they can jump in after glycolysis in the case of proteins and fatty acids, or glycerol, glucose, and some amino acids can jump in at the end of glycolysis, depending on what they convert down to. Okay, so you need to be able to look at that chart and identify where those different foodstuffs are entering the energy pathways. Cellular respiration is not always aerobic. Many organisms survive in the absence of oxygen. Glycolysis produces ATP without requiring oxygen in this case. So aerobic cellular respiration, NADH, gives up its electrons in the electron transport chain, generating the NAD plus need for glycolysis. The electron transport chain requires oxygen. So what can cells do in an anaerobic situation? They have alternate pathways called fermentation or anaerobic respiration. Many prokaryotes use anaerobic respiration. Anaerobic respirations include the cell cycle and the electron transport chain. Electron transport chain uses electron acceptor molecules other than oxygen. Different electron acceptors allow for less ATP production than oxygen. So, other than oxygen, they can use nitrates, sulfates, carbonates. Many prokaryotes and some eukaryotic cells use fermentation. There is no Krebs cycle or electron transport chain in fermentation and no ATP production. Fermentation simply allows glycolysis to continue producing small amounts of ATP. Fermentation also regenerates NAD+. Fermentation uses pyruvate to oxidize NADH and generate NAD+. The pyruvate is converted into alcohol, lactic acid, or other byproducts. Microbes carry out alcoholic fermentation. In alcoholic fermentation, NADH produces pyruvate to ethanol. NAD+ is recreated. That's what happens in beer fermentation. Lactic acid fermentation is another pathway. In lactic acid fermentation, NADH reduces pyruvate to lactic acid. NAD+ is recreated. During fermentation, only some of the energy from glucose is extracted. The oxidation of a glucose molecule only leads to ATP. So what is the main advantage of fermentation over aerobic cellular respiration? A, fermentation generates ATP even if oxygen is not present. B, fermentation generates more ATP per glucose than aerobic cellular respiration. C, fermentation does not generate toxic byproducts such as CO2. D, fermentation gets rid of pyruvate, which would otherwise accumulate in the cell. The answer is A. So even if oxygen is not present, the continued generation of ATP can happen just at a much slower rate. Okay, so the advantages of anaerobic versus aerobic cellular respiration. Aerobic respiration produces much more ATP, but if oxygen is not available, ATP can still be produced more slowly with fermentation or anaerobic respiration. Okay, photosynthesis and respiration are related. They are complementary. They're connected in many ways. Water, oxygen, carbon dioxide, and sugars are all part of their reactants and products. They're just reversible reactions. They both involve cyclical energy creating and energy using processes. They both occur in the cell. Both chemical processes arose in unicellular organisms over 3 billion years ago. Photosynthesis may have evolved from glycolysis, since some Calvin cycle reactions are the reverse of glycolysis reactions. Since virtually all cells carry out glycolysis, is probably the first of these energy pathways to arise. It's the most common of the processes between organisms. Okay, so photosynthesis and respiration compared. So photosynthesis is done by autotrophs, or plants, photosynthetic plants, or algae. Food is produced. Energy is stored as glucose and other sugars. Light is required. Water is consumed. Carbon dioxide is consumed. Oxygen is released. On the other hand, respiration consumes food and releases energy from glucose and other food molecules. Light is not required. Water is released, CO2 is released, and oxygen is consumed. Okay, so they're not the exact reverse of each other. The chemical reaction summaries are the reverse of each other. Glycolysis is the most similar step between the two because some parts of photosynthesis are the reverse of glycolysis. So they're not exact opposites, but there are several similarities and complementary aspects. Okay, the philodendron Limoscens plants warm their flowers using an alternate pathway into the electron transport chain. The electron transport chain prematurely transfers electrons to oxygen. Instead of extracting the remaining potential energy in the electrons, the energy is lost as heat, which warms the flowers. Benefits of the plants: the beetle pollinators are attracted to the heat and pollinate the plant. So both the beetles and the plants benefit. Warm beetles use less energy. The energy they save can be used for finding food or mates. The hot flowers therefore increase the reproductive success of the beetles and the plant. All right, thank you. That's chapter 6.